Good morning, dear students. It's my pleasure today, after we have finished the sector where we studied together the CNS active agents. It's my pleasure today to start with you a new class of medicinal agents that we can identify that this class is belonging to the otacoids. Such a class of drugs will include two big sectors. The first one will be considered with the chemistry of inflammatory mediators and prostaglandins, and hence the drugs acting as non-steroidal anti-inflammatory drugs. And at the same time, after we finish this part, we are going to study the chemistry of histamine and related drugs. So today, inshallah, we are discuss the drugs used to treat pain, inflammation, and arthritis, and this will be in part 1 of the lesson. Next week, inshallah, we are going to understand more the different classes of this category of drugs.
Pain and inflammation mediators is the first point we have to understand before understanding or mastering the part of drugs treating pain or inflammation. What is pain? Pain is a sensation of douleur, that might be rising due to physiological disturbance, inflammatory causes, or even neuropathic cause. Inflammation, as we all know it, it starts by an initial injury. This injury might be a burn, whatever was the provocation, followed by release of inflammatory mediators. Inflammatory mediators, as you have already taken in pharmacology, they can be prostaglandins, histamine, serotonin even. This is followed by vasodilation, increased vascular permeability and exudation, and this two signs, vasodilation and increased vascular permeability and exudation, they are seen as redness and oozing at the traumatic site. Then this is followed by leukocyte migration and then proliferation of connective tissues. Now, what is the role of chemical mediators in inflammation? Prostaglandins, thromboxane, prostacyclin, and leukotrienes are the main chemical mediators precipitated in inflammation and increasing the inflammatory process. Let's start with prostaglandins. Prostaglandins, they have their own very important role in inflammation. These chemical compounds are naturally derived. They are composed of 20 carbon, their precursor and their prototype structure is composed of 20 carbons. 20 carbons is referring as eicosanoids. Eicosa refers to the number 20. So when we are going to say eicosanoids, so these are compounds having 20 carbons. Count them, please. You are going to find that there is a cyclopentane ring including the carbon number 8, 9, 10, 11, and 12. They are all derived from mammalian tissues. Their precursor is a polyunsaturated fatty acid, We are going to see this in the coming near slides. They are pharmacologically named as otacoids. They represent a group of the otacoids. The polyunsaturated fatty acid that they are derived from is a part of the product of membrane phospholipids. To understand exactly the pharmacophoric groups of the prostaglandins, let's study the general structure of prostaglandins. As I said before, the cyclopentano ring is almost at the middle of the compound. We have two aliphatic chains from each side of the ring. Please look to position 15. We are going to find an alpha OH group that is directed back side the plane, and this is pharmacophoric for the activity of the prostaglandins. We are going to find here at position number 13, between 13 and 14, we are going to find a double bond that is trans in its geometrical isomerism. The two side chain around the cyclopentano ring are also showing or demonstrating a trans configuration. Analyzing some of the prostaglandins, you are going to find sometimes written PGE, PGH, PGF. What is these letters representing? These letters are defined according to the oxygen substitution or level at position 9 and 11. So if you are going to find one oxo function, double bond O, as you see here on position number 9, and an alpha OH at position number 11, this is the type E. If two OHs directed backward as alpha substituents at 9 and 11, this is type F. Sometimes we are going to find a number on the PG prostaglandin. So PGH1, PGH2. The number 2 is referring to an additional double bond that is present between C5 and C10, and its nature is cis. It will be geometrically a cis double bond between position number 5 and 6. Coming to the biosynthesis of prostaglandins. Prostaglandins are derived from a precursor that is an unsaturated fatty acid called arachidonic acid. This arachidonic acid is comprising 4 cis double bonds. As you can see the structure, specifically it's the precursor of the 2 series. The 2 series means that those prostaglandins possessing 2 double bonds. As well, arachidonic acid is a 20 carbon unsaturated fatty acid. It represents the precursor of all these class of 2 series prostaglandins present in our bodies. To trace chemically the synthesis of different types of eicosanoids, starting from the cell membrane phospholipids, the phospholipase A2 is capable to generate arachidonic acid having its 4 double bonds. Then comes the role of the cyclooxygenase isomers COX-1 and COX-2 to release or to run a cyclization reaction and an oxygenation reaction producing the parent prostaglandin, which is PGG2. PGG2 will be further differentiated into PGH2 that will be differentiated to PGE2 and PGF2. here, if we want to apply the way to name the prostaglandin, so please have a look on position 9 and 11 to understand that E and F are two different oxygenated forms. Please look to the number 2 to identify the presence of an extra double bond. So now, this is the way to produce the different prostaglandins, starting from the parent arachidonic acid to the PGE2 or PGF2 alpha. At the same time, lipooxygenase LOX enzyme will be able to generate the precursor of all leukotrienes, and we can find that by thromboxane synthase and prostacyclin synthase, we can obtain thromboxane and prostacyclin. Okay, now all these mediators are present. Normally, corticosteroids can inhibit the activity of phospholipase A2, and non-steroidal anti-inflammatory drugs are able to inhibit the activity of cyclooxygenase. So, using a non-steroidal anti-inflammatory drug will prevent the cyclooxygenase enzyme, right, leading to the accumulation of arachidonic acid. So arachidonic acid instead of being synthesizing prostaglandins, it will be accumulated, and the only way it finds is to be treated with lipoxygenase to produce a large amount of leukotrienes. So please keep into consideration that using non-steroidal anti-inflammatory drug preventing or inhibiting cyclooxygenase from cyclization of arachidonic acid will precipitate a large concentration of leukotrienes that is not suitable for asthmatic patients, a case that is not suitable anyway for an asthmatic patient. But for asthmatic patient, it's better to use corticosteroids instead of non-steroidal anti-inflammatory drugs, so that we prevent the cycle or the synthetic pathway from an early step by inhibiting phospholipase A2. This way there will be no prostaglandins, at the same time no accumulation of leukotriene. Different types of prostaglandins and related mediators are having different types of physiological responses that helps in a way or another in keeping the homeostasis of the body, its temperature, blood vessel tone, bronchial tone, and so on. So some of them, and I don't want you to study which of which is doing what exact physiological activity. Some of them are mediating pain, okay, and they have a vasoconstrictor effect. Others are mediating uterine contractions. At the same time, they have an effect on smooth muscles. They have a stimulating effect on smooth muscles, and they can, some of them, as I told you, elevate the body temperature. On the other hand, they have vasodilator properties on some vascular tissues and constrictor effect on different tissues at different sites. Some of them are precipitating relaxation of the bronchial and tracheal muscles, while others are causing their contraction. تحديدا PGI2 is inhibiting gastric secretion. That's why they have a cytoprotective effect. They have a role in keeping the integrity of the gastric mucosa. That's why, please and pay attention, when we are saying that non-steroidal anti-inflammatory drug is having a side effect, that is, it can produce heartburn or gastric acid secretion, uh, um, uh, no protection against the gastric acid secretion. This is the main cause of this. From another point of view, it has another way to protect the gastroduodenal mucosa by stimulating the secretion of alkaline mucus, and this has a role in protecting the gastroduodenal mucosa. These are not the only functions of prostaglandins, but we can find, for instance, that thromboxane is having a potent vasoconstrictor, and it induces platelet aggregation, while on the other hand, prostacyclin, which is PGI2, is a potent hypotensive and having an inhibitor effect on platelet aggregation. This counterbalancing effect is maintaining the homeostasis of blood fluidity and integrity. On the other hand, leukotrienes, they are acting as potent hypotensives and potent bronchoconstrictors. So from the detailed functions now I told you about, we can realize the counterbalancing effect and the homeostatic effect that different prostaglandins are providing the body with. As I told you that leukotrienes accumulation is precipitating sensitivity reaction and inflammatory reactions. That's why it's not favored, it's not in favored to treat asthmatic patient with non-steroidal anti-inflammatory derivatives. This is a protective effect for the asthmatic patient to treat its inflammation with corticosteroids rather than non-steroidal anti-inflammatory drugs. As we have different types of prostaglandins, so we can expect the presence of different types of prostaglandin receptors, and according to their substrate, according to their agonistic prostaglandin, we can differentiate prostaglandin receptors as their names by giving, for instance, PGEs, the receptor to be E receptors, giving prostaglandin type F receptor, giving thromboxane receptor TP receptor, giving prostacyclin receptor IP receptor name. Their activity is governed by the G-coupled protein. So they are considered as G protein coupled receptors. They lead to the release of phospholipase C adenyl cyclase and increased levels of cyclic AMP and IP3 levels. One of the important factors that should be kept into consideration is the metabolic pathway of such a structure. Prostaglandins are obeying to various oxidative and reductive pathways upon metabolism. At position number 15, the OH, the alpha OH, is undergoing oxidation to be converted to a ketonic function, being initially as a secondary alcoholic function. At the same time, the double bond present between 13th and 14th position is undergoing a reduction into the saturated compound. There is a cleavage at the alpha side chain separating the acidic part. All these are weak points in the structure of prostaglandin, and they represent labile functions in metabolism of prostaglandins. That's why when we are intending to produce certain prostaglandins to be available in the market, these concepts of metabolism should be manipulated and should be protected so that we can obtain an effective prostaglandin. After understanding the weak points and threats that might a prostaglandin face during its pharmacokinetic journey inside the body, there are some prostaglandins that are approved for human clinical use. The prototype of all prostaglandins is the dinoprost. We can find our pharmacophoric functions present as we have seen. We have the oxygenated functions at position 9 and 11 that permits this prostaglandin to be an F prostaglandin type. There is an extra double bond, an cis extra double bond present at the alpha side chain. Please have a look on this R at position 15. That is substituting the same position where the alpha OH is present. This R, if it's an H, this is dinoprost, the prototype. If it is methylated, this will be the carboprost having a methyl function. The presence of this methyl function is protecting the alpha OH from the oxidative conversion into the ketonic form. So we can find this carboprost as a protected product against metabolic threats. At the same time, the PGE1 derivative, the alprostadil, please look to it. It is apparent to us as a PGE derivative because of its oxidative functions at position 9 and 11, but it seems to be non-protected at all at its 15 position. Have a look to this ester prostaglandin, that its carboxylic function is esterified with methyl alcohol. This is an E-type, and its OH is not present at the 15th position. It is present at its 16th position, being also neighbored by a methyl function. This is the mesoprostol, being a methyl ester. So we can deduce that this final product, this final structure that is mesoprostol, is not an active prostaglandins. It needs to be hydrolyzed inside the body to be converted to the active acid. At the same time, the OH present at position number 16 is fooling the enzyme responsible for the oxidation. That's to say that this OH is protected. It won't be oxidized as in case of dinoprost. According to what we have said, we can deduce that we can overcome the metabolic oxidation of the 15th alpha OH by either by either introducing a methyl that protects it sterically. It will be, the oxidation will be hindered, and we can deduce again that we can ameliorate the pharmacokinetic properties and lipid solubility of the prostaglandin by its esterification at the carboxylate to be a prodrug that is active. Prostaglandins have many therapeutic uses. We can find in the market some abortifacient prostaglandins like dinoprost and carboprost. Some of them, like misoprostol. Misoprostol is a synthetic prostaglandin, E-type. It is used for its capability to inhibit the secretion of HCl. That's why it is used in patients with gastric ulcer. Another medicinal use of prostaglandins is to maintain open ductus arteriosus, like alprostadil in neonates, and it treats male impotence. Coming to the non-steroidal anti-inflammatory drugs, after good understanding of the prostaglandins and how they initiate pain and how they are synthesized biologically, and how many drugs are present in the market having the different physiological effects that prostaglandins possess, it's time now to study the non-steroidal anti-inflammatory drugs. Non-steroidal anti-inflammatory drugs are sharing a common mechanism of action, which is inhibition of cyclooxygenase, either 1 or 2. Cyclooxygenase is the enzyme responsible for cyclization and oxidation reaction of arachidonic acid to convert it into the parent prostaglandin. Some antipyretic drugs related to non-steroidal anti-inflammatory drugs, they exert their action mainly by having a CNS effect, acting at the level of the hypothalamic thermoregulatory centers. They also have an antagonistic effect at the level of bradykinin, which is a potent pain inducer. In the coming two slides, we can know the different painful conditions where non-steroidal anti-inflammatory drugs are indicated. We have to keep into consideration that non-steroidal anti-inflammatory drugs, they have their symptomatic relief. They are not typical treating agents. They are used in acute gout, not to treat gout, rather to alleviate the pain accompanying the gout condition. It is also used in rheumatoid arthritis, osteoarthritis, and some spondylitis. Before the development of the new scientific technology, the anti-inflammatory receptor was hypothetically determined according to the prototype of all anti-inflammatory drugs, which is indomethacin. Indomethacin, according to its core structure, it helped the researchers to understand the topography of the inflammatory receptor. They identified, according to a complementary structure to indomethacin, that the anti-inflammatory receptor is composed of a cationic site able to accept the carboxylate anion, a flat area in order to accept the indole ring, and out of the plain trough that is able to host the benzene ring of parachlorobenzoyl group of indomethacin. By time and by the newer technology, they realized that this hypothesis was exactly confirming the structure of the pocket, the active pocket in cyclooxygenase enzyme. Cyclooxygenase 1 is composed of 599 amino acids. COX-2 is composed of 604 amino acids. They have almost similar length, having almost the same number of amino acids. The catalytic site of both isomers are identical. Some minor differences are present between the COX-1 and COX-2. The remarkable difference is a difference that causes the selectivity of COX-2. The isoleucine amino acid present in position 434 and 523 in COX-1 is exchanged for valine in COX-2. That's to say, COX-1 is having an isoleucine residue at these two positions, 434 and 523, while COX-2 is having instead valine. The selectivity is arising from the smaller size of valine in COX-2, which allows the bulky inhibitors to access the pocket site of the enzyme, while isoleucine having a bigger size with a longer side chain in COX-1 sterically inhibits the access of the traditional types of non-steroidal anti-inflammatory drugs. The COX-2 inhibitors are also different, having no anionic part. That means that they are not binding to the arginine 120 that is responsible to host the carboxylic group of arachidonic acid. Another teeny difference is that there is a sequence of 17 amino acids are lacked in COX-2. They were switched from the N-terminus to the C-terminus. This is sure in comparison to COX-1. Coming to aspirin, is covalently inhibiting COX-1 because it's able to acylate the serine residue in COX-1. That's why it has a specific mechanism in fitting the receptor through covalent interaction and acylation. This serine in COX-1 is present as serine 530, while in COX-2 it is serine 516, and this is of course because of the switching of places of the 17 amino acid residue as we just mentioned. Analyzing the differences we said in the last two slides, we come to a conclusion that there is no big differences between the two isomers of the enzyme COX-1 and COX-2 from the chemical point of view. Rather, the difference is mainly physiological rather than structural, because we can identify that COX-1 is responsible to produce the prostaglandins that are involved in the normal physiological responses, the normal cellular activities concerned with gastric mucosa protection, managing kidney functions, whereas COX-2 is the entity involved in production of inflammatory prostaglandins. That's why the new research and the new selectivity and drug design is focusing on developing drugs that are more selective and specifically acting on COX-2 enzyme. As if we have two types of COX, a good COX and a bad COX. The COX that is present in our body to stabilize the body function is the good one, which is COX-1, and the bad COX is mainly producing those prostaglandins, producing inflammation and pain conditions. When we start to classify non-steroidal anti-inflammatory drugs, we settle to a therapeutic classification that starts with the antipyretic analgesics, then the non-steroidal anti-inflammatory drugs, then those acting as disease-modifying antirheumatics, and they are abbreviated as DMARDs, DMARDS, and finally those drugs treating gout. As for the therapeutic classifications, we start with the antipyretic analgesics. The prototype of this category of analgesics is paracetamol. Paracetamol is having another name that is acetaminophen. Both names are describing the presence of an acetyl function allocated on an amino group. This amino group is actually on a phenyl ring, and at the para position there is an ol, a hydroxy function. The mechanism of action of such a compound, which is paracetamol, is by inhibition of prostaglandin biosynthesis in CNS. That's why they have their antipyretic activity. We all think that paracetamol is a very safe drug, but is this true? The metabolism and fate of paracetamol is dictating the conjugation by O-sulfate or O-glucuronide. So it is conjugated as O-sulfate or O-glucuronide conjugate, and this way the drug is coming more hydrophilic and the metabolite is easily excreted. But there is a minor fate, a minor chemical conversion is present by the conversion to N-hydroxyamide. The N-hydroxyamide is then converted to another toxic metabolite, which is acetimidoquinone. So to describe what we have said in chemical equations, we can observe that in children the main metabolic pathway is by O-sulfate conjugation, and in adults it is by O-glucuronide conjugation. A minor fate or a minor metabolic pathway is including the conversion into N-hydroxyamide derivative, where there is an oxidative action on the nitrogen, replacing its hydrogen with a hydroxy function. Further, this group is replaced by an quinone structure, what we are calling acetimidoquinone. This acetimidoquinone is a hepatotoxic and nephrotoxic metabolite. How this comes? It is able to interact with glutathione and deplete the body from the glutathione. We all know that glutathione is one of the defensive mechanisms in our body against oxidative stress. So glutathione will interact with the acetylmediquinone, and the body is able to get rid of this minor metabolite. But in case where patients are continuously administering paracetamol in excessive doses, there is a depletion of glutathione, and the only pathway that is available is that this acetylmediquinone will interact with the SH groups of hepatic proteins, causing covalent interaction with the hepatic protein and their necrosis. N-acetylcysteine is used as an antidote for acetaminophen overdoses. There are some companies that are incorporating N-acetylcysteine in paracetamol formulation in order to protect the body against any possible residues of acetylmediquinone, reserving the body glutathione system. The chemical classification of non-steroidal anti-inflammatory drugs is very specified into salicylates, aryl arcanoic acids, and aryl anthranilic acids, 3,5-pyrazolidine diones, oxycams, and selective COX-2 inhibitors. Salicylate is having a prototype that is acetyl salicylic acid, aspirin. Aspirin is interacting irreversibly with COX by covalent interaction at the level of serine 530 in COX-1 and at the level of 516 serine in COX-2. Tackling the thermoregulatory centers in the hypothalamus, aspirin is having an antipyretic effect by blockade of prostaglandin synthesis. Low doses of aspirin are used to inhibit the thromboxane production in platelet, so aspirin is having an anticoagulant effect in low doses of 60 to 80 milligrams per day. The salicylates have as pharmacophoric group the carboxylate anion that is able to interact ionically. So the anionic group is a pharmacophore of this structure. Salsalate, which is salicyl salicylic acid, is a derivative of aspirin. It is an ester formed between salicylic acid, ester of salicylic acid. Of course, this ester is readily hydrolyzed after absorption, causing less gastric upset than aspirin and being relatively insoluble in stomach. It is mainly absorbed in small intestine. The 5 position of salicylic acid, its substitution increases the anti-inflammatory activity. Let's see this structure, diflunisal. It has at the 5 position a difluorophenyl function. Of course, this ring is adding to the lipophilic properties of the salicylate derivative, and it increases its bioavailability.